So, if everybody's ready, we're ready for the next session. I'll just introduce myself, by the way. My name's Jonathan Milner, and I founded ABCAM a long time ago, 1998. And I'm obviously enormously proud of the work we're doing in this field, and it's really dear to my heart, because the reason I founded ABCAM in the first place was because I couldn't get access to decently validated antibodies. Immensely frustrating. Spent 2 years working with an antibody that wasn't specific. You'll guess the vendor for that one. They're not here today, by the way. So we have two really amazing speakers coming up. We've got Professor Lars Björk, and Lars is currently heading up the Human Protein Atlas and working at Karolinska Institute and the Royal Institute in Stockholm. So he's going to tell us all about his work, and then we've got Dr. Alejandra Solache from ABCAM to tell you about what we're doing in knockout validation. So I will try to keep us to time, and over to you, Lars.
Okay, thank you. And thank you, Jonathan, for my promotion to heading the whole HPE project. That's really kind of you. And I'm really happy it's on video also. So I'll be talking about the Human Protein Atlas and how to confirm subcellular protein localization. And I work, as Jonathan said, both at the Royal Institute of Technology. I also have an employment at the Karolinska Institute. We're all based in the same building, actually. And it's called the Science for Life Laboratory, which is a national initiative where core technologies is available throughout scientists around the country, primarily. So, and I just realized when I gave this talk that actually antibodies are the reason I'm a scientist. Because a little bit earlier than 98, January 1985, I did a internship as a 17-year-old at my mother's choir friend who was working at the State Bacteriological Laboratory in Stockholm. And luckily, Köhler and Milstein just had been awarded the Nobel Prize. And there was a lot of articles in Swedish about immunity and antibodies. So I could easily read myself through this and realize that immunology was really a cool thing. And actually, at that time, I did immunofluorescence with monoclonal antibodies. It was really cool. And then it's always regarding the sort of the build-up of the commercial use of antibodies is always, now it's 40 years ago since the NRDC recommended that antibodies had no commercial value and we should not patent it. And it's kind of one of the worst refusal letters I think I've ever. The other one is that Pharmacia Biotech, when my professor Peter Perlman and Eva Engvall, who discovered the ELISA method, went to Pharmacia to ask them if they could co-patent it. They said, no, we have the RIA and it's fine. So that's the other one. But this is worse. Luckily, they were awarded with the Nobel Prize. Okay, so the Human Protein Atlas project, it's actually directed by Matthias Ullén, and it started 13 years ago as an initiative post-Hugo. So the notion that the genes isn't the man, it's the proteins who's the man, and the difference between us and other chordates are perhaps just the level of proteins and in which cell they are expressed. And it was funded and is still funded by Knut and Alice Wallenberg Foundation. So that's ongoing funding for now 13 years, which we're very happy of. There are more than 100 people working in this project, and altogether we've done more than 1,000 person-years FTEs. So it's an impressive endeavor. And the idea behind the project is to characterize spatially the proteome in tissues, in cells, and in pathological material. We do that so the spatial proteomics could be used for many different things, and compartmentalization is, of course, one aspect of how protein works. and it could provide clues for function, which tissue. It can provide clues for adverse effects. Why does a drug that's directed towards liver protein all of a sudden give a headache? Well, maybe the protein is also expressed there, or the metabolites are winding up there. So that's another aspect of compartmentalization and spatial proteomics. So we are doing immunofluorescence, we're doing immunohistochemistry in tissue and organs, and we actually RNA sequenced both the tissue of interest, but also all the cell lines that we're using. So they are characterized for their mRNA expression. 44 normal tissues have been sectioned into tissue microarrays. We have more than 20 cancer cell lines, and of course all the 48 cell lines that we use for the cell atlas. What's special about this project is perhaps sometimes science starts with the interest of a specific protein. But here it was proteomics, and we wanted to characterize as many proteins, if not all, as possible. And in order to do so, since we didn't know the proteins at heart, we need to start with reagents. And actually, I think they tested more than 20,000 different commercial antibodies in the project. But in the end, it turned out to be an internal endeavor to do actually antibodies within the project. So this is the HPA workflow. And a part of this is to select a good epitope. We call it protein sequence signature epitope. And what we're doing is that we're selecting as unique sequences as long as possible in order to get the folding correctly. And then we do a protein factor on that, inject it into rabbits, and then we are testing them again, and then we're separating the antibodies in affinity column based on the same antigen. And then we're doing a protein array to see that the antibody of interest only detects that sequence. And then we go further, we're doing immunohistochemistry, we do a Western blot, and we do immunofluorescence using confocal microscopy to detect it. So that's the workflow. So in generating the antigen, we have different tags we put on. We clone it into E. coli, we DNA sequence and verify the clones. We do purification, MS verification of the antigen, and then we do immunization in rabbits. And then we purify it on the same pressed in a column, so that becomes an affinity purified polyclonal antibody. And then we do these quality assurance with the pressed array, as we call it, where we have over 48 array validated antibodies today, and we put them on 3 default protein fragments in 21 subarrays. So that's one way of screening the specificity or the multivalency of the antibody. And if it doesn't approve here, then we cannot use it in our assays, because then it will bind many targets. We can actually use this in research as a research tool also to look for autoimmunity against human proteins. So that's another part of the project where we look at serum samples from patients and whether they have activity against endogenous proteins. The tissue microarrays have today generated more than 13 million annotated, meaning described images in various tissues. And we do, it's on the level of single cell resolution. We have a slide scanner, and then we have pathologists that have worked with these images and looked at them and verified their stainings. And we do this as comment then. So the first true draft of the tissue atlas was released 1.5 years ago in December, January. And it contains spatial proteomics data with 44 tissue types and RNA sequences. And we were able to do subproteome characterizations of all the proteins expressed in this. So it was about 80% of the proteins characterized by this methodology. If you look on and then what interesting again is that all tissues are very similar to one another. More than 50% are expressed in all tissues. And there are a few organs that stick out. The brain sticks out. And then there's the testis sticks out and has almost 1,000 unique genes expressed in this. And that was very fun for the media, of course, because they can have the connection. But in reality, of course, testis is a very unique organ with very unique proteins in order to deal with the meiosis and spermatogenesis. So most important conclusions, almost half of the genes expressed in all cells of the human body. Very few proteins were unique for one tissue. And if you look on the druggable proteome, we're only talking about percentage here that actually are used as drugs today. So there's more to do. Another part of the Human Protein Atlas is that we've been looking specifically into brain, and then it's very difficult to use human samples if you want to look at where proteins are expressed on a whole brain issue for obvious reasons. So many of these proteins are, of course, very similar in the mouse brain. So we have a project we call the Mouse Brain Atlas, where so far we've done about 100 genes, characterized them on brain level. And it's a very nice interface with a virtual microscope here. So that's worthwhile looking into. It's headed by Jan Mulder, who is only employed by the Karolinska Institute. I personally work in the Cell Atlas group, and it's run by Emma Lundberg. And we have been using the next level of resolution, being confocal microscopy, to characterize where in the cell these proteins are. If you know which tissue they are, where are they then expressed in the cell? And we do this by basically traditional confocal microscopy, but at as high resolution as we can possibly do with normal. So we actually are publishing images with Nyquist resolution in the 63x oil immersion lens. So that means around 1700 pixels resolution. And the actual raw images here are 16-bit grayscale. And if you look on the atlas, they're actually enlarged to 2048 pixels by 2048 pixels. So there is an enormous amount of data. When we do one plate for imaging, we generate about 250 gigabytes of data. And then we characterize them in an annotation procedure for different subcellular compartments. It could be the nucleus, it could be the Golgi, it could be nucleoli, cell membrane, etc. And that's done by an annotator who doesn't know what the target is. And then we review it in a later stage, look at double stainings with another antibody, and then we also check the literature before we're publishing it as an annotated. So the workflow looks like this. We select our panel from about 30 human cell lines and the ones that are truly expressing. So that's another way we're doing is that we have all these cell lines sequenced based on their RNA, so we know whether they should be expressing the genes or not. We have an automated system because this is thousands of stainings, and we do about 1,500 to 2,000 images per week using the system. And then we do all the stainings in a robot, and then we do a confocal microscope system. And we also can run this automatically overnight with auto exposure scripts that we've done. And then we do feature extractions, and we have projects with trying to investigate where in the cell cycle it is based on different morphology of the cell. So all cells are stained for DAPI. All cells are stained for microtubules. All cells are for calreticulin or KDEL as an ER marker. And then we have the analyte that we reference to. So that's the way we do it. And we can then look at the analyte is always in the green channel, and it could be cytoskeleton, in microtubules, actin filaments, etc., intermediate filaments, and they have their own characteristics. And then we do present the data like this. And of course, again, many antibodies are actually localized, or proteins are localized in multiple compartments. So that's something we see again. So now we're annotating in a refined way with all these different subcellular compartments. And the antibody and antigen information is displayed on the website. So you can actually see if there's any other transcripts, the source of the antibody. If you look here, you can also see what the antigen was, where it was in the protein sequence. So these are the prests and the number on the press. So there's very easily displayed where in the protein these antibodies were. So that's a part of the validation procedure in defining if this is a doable sequence or not. So the current status is that we have made about 57,000 sequence verified clones of antigens expressing these prests. 42,000 of them were verified by mass spectrometry, and 49 were actually in the pressed array. We've done about 25,000 antibodies with immunohistochemistry and about 30,000 antibodies in cells with immunofluorescence. And all immunofluorescence cells are done by 3 cell lines in order to get a good annotation. And in the end, this is what we are experiencing at the moment. So we have 11,000 genes that we characterized on a subcellular level. And the aim is to go for another 1,000 or so to the release that we're going to present in December at the American Society for Cell Biology, where we will release the new version of the subcellular atlas that we will then call the Cell Atlas. So how do we verify all these stainings that we're talking about here? And I don't need to show these, they're all already been shown. Antibodies, of course, are inherently a binder that is designed to bind many things initially and then to refine itself through affinity maturation and also boosting and everything. But there can always be a problem with cross-reactivity. And it could bind through the paratope-epitope interaction. It could bind non-target proteins. It could be a variability of the antibody. We talked about batch-to-batch variations, both in monoclonal, but also polyclonal, of course, or even huger batch-to-batch variation. Even if you inject the same antigen in another rabbit, it's going to be very different. Wrong application. I mean, if you have designed a peptide to be totally denatured, it will perform possibly well in Western blot, but probably not well in immunocytochemistry. So, and hopefully the specific binding is in the nanomolar range, whereas off-target binding should be, in principle, in the lower affinity range, which means that if your protocol is having a buffer that's allowing this one to bind but not that one, then maybe you can use this antibody anyway, because it's in this application it performs well. So I think that's very... Then, of course, you always have the noise that we have in our antibody. It could be Fc interactions. It could be very sticky antibodies. We try to deal with that in our protocol, of course. Examples of application dependency is that in immunofluorescence, using this antibody against percentrin, we get a very nice centrosome staining. But in Western blot, there are many bands. So it doesn't really suit the way you use it. And actually, this is a problem for us dealing with immunofluorescence, because in our workflow, Western blot is one of the bottlenecks it needs to go through. So what do we do in the HBA as an antibody validation key? We compare to other experimental evidence from literature. We compare with RNA sequence data across the samples that we're staining. We do multiple antibodies targeting different epitopes of the antigen. We do siRNA-mediated gene silencing, and we do co-staining of cells expressing GFP tracked proteins. So a workflow for siRNA could be like this, where we're actually doing a knockdown, and we do image analysis, and then we do statistical analysis, and then we have a scrambled siRNA as a control. So this is one example where this staining was completely abolished by the knockdown by the siRNA. We also done about 100 genes or more, actually 300 genes with GFP tag. And here it's very important that the antibody that you're interested in validating also binds the GFP tag protein. But it should also bind the endogenous protein. So you should always have that, not only do the GFP and the antibody in this model system, but in control cells where you don't have the construct. And preferably you should also express this in a level which is endogenous, because otherwise you can get into artifacts. So we do this with a collaboration with Anthony Hyman in EMBL. So conclusion, start with the quality of the specimen. I haven't talked about that, but in reality it's really the tissues and cells that we're dealing with that maybe are the downfall. And that doesn't come to the vendors or the people providing the antibodies, come to us dealing with our samples. And you need to optimize your method, of course, but you need to validate it application specific. And then you can strengthen your data with an orthogonal method, a non-antibody based method like mass spectrometry or sequencing. So this is all now proposed and published a week ago in this International Working Group for Antibody Validation paper that came out, where they present that we should have a genetic strategy, an orthogonal strategy, an independent antibody strategy, expression of tagged proteins, and also capture of MS that we will hear more about tomorrow. And if we do one of these pillars, then it's a good control. But there are other controls, and I think a lot of people talk about flow cytometry. We never done any of this when we were doing flow cytometry in the 90s. We did isotype control and we did adsorption. And it shows the epitope and the paratope interaction in the absorption function. But of course it cannot say whether you have off-target binding. So that's the downfall. But most of the data in flow cytometry hasn't used any of this to my knowledge. The traditional control is here. But flow always have an isotype control. Immunohistochemistry quite often forgets that and only use the secondary antibody. Okay, so consideration for development of antibody validation, application specific, adaptation to context specific validation, and development of validation strategy should incorporate community input. And what we're talking here is really the producers and the users coming together for validation antibodies and sharing this information. And it's a bit problematic because we also know that sometimes these antibodies from different vendors are the same antibody because they've been sub-licensing to one another and they don't want to tell that. But I think one initiative that is a poster outside is what Tuval from SciLife Lab is presenting, the knockdown initiative, where scientists can get rewarded from vendors if they validate their antibody in this context. So I will like to finish up with acknowledgement. This is the group of Emma Lundberg where I'm a part of, and I'm the lab manager, so I'm running all these production things. We have, of course, other people in the HBA, and we have our collaborators. And also like to, again, acknowledge the people who's been supporting us coming here, because these are actually the vendors that will be doing these community-based validation. And if they're into it, then it's going to happen. Otherwise, we have to develop our own antibodies. Okay, thank you.
Thank you, Lars. I think you'll agree it's an enormously impressive project and on time. That's just a miracle. So we do have time for some questions. So I'll open up the floor. Anybody want to?
Hi. So, do I understand correctly that Western blots are now no longer to be used for validation? Is that what you're proposing? Because I didn't see that in any of the pillars, and I didn't quite understand your comment where you said that this antibody gives multiple bands in Western blots, and yet it gives a good signal in immunofluorescence. I mean, how do you know that some of that immunofluorescence isn't due to recognition of some of those other bands? Yeah,
then I have to go back to the raw data, but I mean, I think Western blot is indeed one of the pillars. I'm not a member of the IFGAV group, so I cannot stand and talk for them. I'm just a bystander and see what they're doing, but they're my bosses. So, but I think that if you look back on what's in there, so is antibody-based. If you want to validate Western blot, you validate it for Western blot. But just because a Western blot is validated doesn't mean it will perform well.
What about the contrary?
So if it performs well, actually, if you have a conformational epitope that's been generated because the antigen in question was a recombinant protein or a long enough sequence, then it will possibly not perform well in Western blot.
No, that's not the question. If it performs badly in Western blot, then I would argue you shouldn't really be using it at all, period. Because it's recognizing other stuff. Are you disagreeing with that?
So then I have to sort of talk. I can't speak for IFGAV in that aspect. I think that if you want to look at a band, this is what I think. I think it's probably not a good idea because you have a problem here. Maybe Tove wants to jump in. But my feeling is that if you have a lot of bands here that's related to other proteins, because first you have to see that it's actually other proteins, then you have off-target binding. And either you can do, as many people, just cut out this band and publish it and say it's difference. That's one way to do it. It doesn't mean necessarily that it doesn't measure differences in that exact protein, but it means that you have off-target binding.
And that's the issue. That's the whole problem with validation, is off-target binding.
Yeah.
We've got time for a couple more questions.
Tove, do you want to jump in and explain better?
I have one question. So about half of the annotations for the subcellular location in the current release are annotated as uncertain, right?
Yes.
So how difficult or easy do you think it is to do definitive validation in immunofluorescence? Application specific.
Yeah, so it probably means then that we have to, if we're going to do this, then we have to use one of the pillars, and we have to sort of, we have to go back and then knock down or knock out. That's one way. We can do GFP tagging. That's one way. We can do another antibody, because sometimes, a part of this uncertainty that we have in validation is that we only were able to find one binder.
But if you have two antibodies binding to the nucleus, do you think that is strong evidence that they're binding to the same protein?
So if they were, if they are from different epitopes, and we know from a sequence point of view that these sequences are not expressed, that we take as an additional validation of the antibody.
But how many proteins are there in a nucleus?
That's 3 questions. We've got time for a couple more.
I'll try to be brief. So it's Corinne Lisa from AstraZeneca. So I had 2 questions, I will just ask one. The first one is whether you've seen some correlation between the protein array screening that you do upstream with your antibody and the final result in IHC, and whether you can, by looking at the way your antibody works in the protein array, predict which result you will get in your IHC result.
So the protein array is done as a quality control issue, so we won't actually release it for immunohistochemistry if it's not performing well in the protein array. So that's how we do it.
Discard it straight away from the...
It would be failed in pressed array as we speak, and then we would not use it.
So is there any workflows in which an antibody could fail in all, so the protein array, the Western blot?
And still get published? No?
Well,
no?
Or be tested by you in immunohistochemistry?
Well, what happens is sometimes that we, since it's different protocols, so it's performing well in the pressed array, but it's not working well in immunohistochemistry. It could still work in immunofluorescence because it's a different protocol.
Great, we've got time for just one more question, please, and then we're going to move on.
Hello. You did make the comment that you've got a lot of batch-to-batch variation, because you are using polyclonals.
Yeah, I'm saying that it's a problem. I'm not saying that we have a lot of batch-to-batch variation, because usually we only have one batch. So there's very few antibodies where we come back, but it do exists.
Okay. So is the protein atlas going to be sort of revalidated with monoclonals?
So, I mean, in those aspects where we don't find suitable antibodies, we are using commercial antibodies, but I don't think we will do initiative of trying to do monoclonal for 20,000 proteins. But I think that when we don't get sufficient amount of data with the current antibodies, then of course monoclonal are interested. But we're also doing, I mean, it's a curation process. So if we get additional antibodies from either from external sources, from collaborators, because we're only doing this amount of proteins. The remaining ones are the difficult ones, so we need more antibodies in order to sort of come to the complete. And then, of course, sometimes in an update in the database, we lose genes that we thought we designed a PREST, it has some kind of affinity to that, and then it realizes it's a pseudogene. Then we need to create that one.
Great. I think we're going to be over time, and that's going to eat into lunch. So I'd like to thank Lars again. Thank you.
Thank you.